Welcome to Intention. And the obvious idea, switch telomerase back on, runs into a problem. The limit on cell division is a genuine defense against cancer. A cell that can divide indefinitely is closer to being a tumor. Most cancers reactivate telomerase precisely in order to become immortal. So this is not a simple lever. The third hallmark is epigenetic alteration, and it is currently the most exciting area in the entire field. Your genome is the same in every cell, but which genes are switched on differs enormously between a liver cell and a neuron. That control is epigenetic, chemical marks, particularly methyl groups attached to DNA, and modifications to the protein's DNA wraps around, which determine which instructions get read. With age, this pattern drifts. Genes that should be silenced become active, genes that should be active get silenced. Cells gradually become less certain about what they are supposed to be. In 2013, Steve Horvath demonstrated that patterns of DNA methylation at specific sites predict chronological age with remarkable accuracy across tissues. These epigenetic clocks are now a major research tool, and later versions attempt to predict mortality and disease rather than simply age. The exciting part came from a different direction. Shinya Yamanaka won a Nobel Prize for showing that 4 specific factors can convert an adult cell back into a stem cell, effectively resetting its epigenetic state to an embryonic one. Subsequent work has shown that applying such factors partially and transiently can restore youthful characteristics to old cells and tissues in animals, including a striking result in which vision was restored in aged mice. This has led to the argument, associated particularly with David Sinclair, that aging is fundamentally a loss of epigenetic information rather than accumulated physical damage, and that the information may be recoverable. I want to be clear that this is a genuinely contested proposition. It is an active and serious hypothesis with impressive supporting experiments, and it is not established, and there are careful researchers who disagree. Full reprogramming causes cancer in animals, which is why the work focuses on partial and controlled approaches. There is no human therapy, and anyone selling you one is not being honest. The fourth hallmark is loss of proteostasis, meaning the systems that fold proteins correctly and clear away damaged ones become less effective. Misfolded proteins accumulate and aggregate. This is directly implicated in Alzheimer's, Parkinson's, and other neurodegenerative diseases, all of which involve protein aggregates. The fifth is deregulated nutrient sensing, and this connects directly to our earlier episodes. Cells have sensor systems detecting how much energy and nutrients are available. The insulin and IGF-1 pathway, a growth signaling complex called mTOR, an energy sensor called AMPK, and a family of proteins called sirtuins. These pathways are among the most reliably manipulable levers on lifespan in laboratory animals. Reducing insulin and IGF-1 signaling extends life in worms, flies, and mice. Inhibiting mTOR with the drug rapamycin extends lifespan in mice, which is one of the most reproducible findings in the field. Caloric restriction, which affects all of these, extends lifespan across many species. The common theme is a signal that says food is scarce, which shifts cells from growth mode into maintenance and repair mode. That is the disposable soma theory playing out biochemically. When reproduction looks unpromising, the organism reallocates toward keeping itself alive. The sixth is mitochondrial dysfunction, and here there is an important correction to make. Mitochondria generate your cellular energy, and in 1956 Denham Harman proposed the free radical theory of aging, that reactive oxygen species produced during energy generation damage cells cumulatively, and that aging is essentially oxidative damage. It is an appealing theory. It launched the entire antioxidant supplement industry. And in its simple form it is now largely rejected. The reason is that the predictions failed. Large trials of antioxidant supplements have not shown the expected benefits, and some found harm, with certain antioxidants associated with increased mortality in some analyses. Some organisms with high oxidative damage live a long time. And crucially, reactive oxygen species turn out to be important signaling molecules, not merely waste. Mopping them up indiscriminately interferes with useful processes, including some of the beneficial adaptations to exercise. The current view is that mitochondrial dysfunction matters in aging, that it is more about impaired quality control and reduced capacity than simple oxidative wear, and that swallowing antioxidants does not address it. That episode is worth remembering as a general lesson. A plausible mechanism plus a supplement is not evidence. The seventh is cellular senescence, and it may be the most therapeutically promising. When a cell is damaged, or has divided too many times, it can enter a state called senescence. It stops dividing permanently but does not die. This is protective, because it prevents damaged cells from becoming tumors. The problem is what senescent cells do while sitting there. They secrete a cocktail of inflammatory signals, growth factors, and enzymes, collectively called the senescence-associated secretory phenotype. That secretion damages surrounding tissue and can push neighboring cells into senescence too. Senescent cells accumulate with age, and in animal studies removing them has produced striking results. Mice engineered to clear senescent cells showed delayed onset of multiple age-related conditions and improved function in later life. This has driven intense interest in senolytics, drugs that selectively kill senescent cells. Early human trials exist in specific conditions. It is genuinely promising, and it is not yet an established human therapy, and some compounds are already being sold to the public well ahead of the evidence. The eighth is stem cell exhaustion. The populations of stem cells that replenish your tissues decline in number and function, which is why healing slows, why immune function declines, and why muscle is harder to rebuild. And the ninth I will mention is chronic inflammation, sometimes called inflammaging, a persistent low-grade inflammatory state that develops with age, driven partly by senescent cells and partly by accumulated damage, and which contributes to essentially every age-related disease. The essential point about all of these is that they are not 9 separate problems. They interact and reinforce one another. DNA damage causes senescence. Senescent cells cause inflammation. Inflammation damages stem cells. Mitochondrial dysfunction generates damage. It is a network, which is why single-target interventions have proved difficult. Before we leave the biology, there are 2 patterns in human aging that are extremely well documented and still not fully explained, and they are worth knowing because they show how much remains open. The first is that women live longer than men, essentially everywhere. This holds in every country with reliable records, across every historical period examined, at every level of wealth, and it persists in populations where behavioral differences are minimized. The gap varies in size, but its direction does not. Among people who reach extreme old age, women outnumber men substantially. The explanations are partial. Behavior accounts for some of it: historically higher rates of smoking, heavier drinking, and more deaths from violence and accidents among men. But the gap remains after adjusting for these, and it appears in other mammals too. Biological hypotheses include the protective effects of estrogen on the cardiovascular system, differences in immune function, and an argument from the sex chromosomes themselves. Having 2 copies of the X chromosome means a damaging mutation on one may be compensated by the other, whereas males have only 1 copy and no backup. Across the animal kingdom, the sex with 2 matching sex chromosomes tends to live longer, which holds in birds too, where the pattern is reversed and it is the males who carry the matching pair and the males who live longer. That is suggestive rather than settled, and it is one of the more interesting unresolved questions in the field. The second pattern is that men are generally physically stronger and more robust at any given age, yet die sooner, and that women spend more years living with disability. So the sexes differ not only in lifespan but in the shape of decline, and that difference is not well understood either. I mention both because this subject is often presented as though the outline were complete and only the details remained. It is not. Something as basic and universal as why half the species outlives the other half does not have a settled answer. Now, the animals that do not play by the rules, because this section is the strongest argument that aging rates are not fixed. The naked mole rat is the outstanding example. It is a small, hairless, subterranean rodent from East Africa. A mouse of similar size lives around 3 years. Naked mole rats have been recorded past 37. More remarkably, a study published in 2018 found that their risk of death does not measurably increase with age. They appear to violate the Gompertz law, which is the closest thing to a fundamental rule in this whole field. They also show remarkable resistance to cancer, associated with an unusual form of a molecule called hyaluronan in their tissues. The Greenland shark has been dated by chemical analysis of its eye lenses to lifespans of several centuries, with individuals estimated at 300 or 400 years old, meaning some sharks swimming today were alive before the Industrial Revolution. An ocean quahog clam collected off Iceland was determined by counting its shell growth rings to be over 500 years old, having begun life during the Ming Dynasty. Bowhead whales live over 200 years, which raises a question known as Peto's paradox. A whale has thousands of times more cells than a human, each capable of becoming cancerous, and lives longer. It should be riddled with tumors. It is not. Large, long-lived animals have evolved enhanced cancer defenses, and elephants, for instance, carry many additional copies of a critical tumor suppressor gene. Hydra, tiny freshwater animals, show essentially no increase in mortality with age in laboratory conditions, apparently because their bodies are composed largely of continuously dividing stem cells. And a small jellyfish called Turritopsis dohrnii can, under stress, revert from its adult form back to an immature polyp stage and begin again, which has earned it the name the immortal jellyfish. It can still be eaten, and its cells are not literally immortal, but it can reverse its own developmental clock. The conclusion from all of this is important. There is no law of physics requiring bodies to deteriorate on a human schedule. Aging rate is a biological parameter, and evolution has set it to wildly different values in different lineages depending on circumstances. That does not mean it is easily changed in humans. It does mean there is no fundamental barrier making the question absurd. So where are humans? Human life expectancy rose dramatically over the last century and a half, and it is essential to understand why, because it is widely misunderstood. The increase came overwhelmingly from reducing deaths in infancy, childhood, and young adulthood, sanitation, clean water, vaccination, antibiotics, safer childbirth. Historical average life expectancies of 30 or 40 do not mean people dropped dead at 35. They mean enormous numbers of children died, dragging the average down. A person who survived to 20 in ancient Rome had a reasonable chance of reaching 60. What has barely moved is maximum lifespan. Jeanne Calment, a French woman, died in 1997 at the age of 122, and her record still stands nearly 3 decades later. In an era when the number of people living into their hundreds has grown enormously, nobody has come close. Whether there is a hard ceiling is genuinely disputed. Some analyses argue for a natural limit around 115. Others argue that mortality rates plateau at extreme ages, which would imply no fixed ceiling. The data at those ages are sparse and the statistics contested. Which brings me to something that most content on this subject will not tell you. You have almost certainly heard of blue zones, regions said to have exceptional concentrations of people living past 100, with lessons drawn from their diets and lifestyles. That work has come under serious and substantive criticism. Research by Saul Newman and others has argued that reported extreme age clusters correlate strongly with poor birth registration, high poverty, and clerical error, and that pension fraud and simple record-keeping failures account for a substantial share of claimed supercentenarians. Some regional records have collapsed under scrutiny. I want to be careful and fair. This criticism does not demonstrate that the associated lifestyle recommendations are wrong. Eating largely plants, staying physically active into old age, maintaining social ties, and not smoking are supported by an enormous body of independent evidence, and do not depend on the blue zones data being sound. But the specific claim, that particular villages have unlocked something unusual, should be treated with considerably more caution than the popular presentation suggests. When the age records themselves are questionable, conclusions built on them are questionable too. So, what does the evidence actually support? Let me separate this into what is well established and what is promising but unproven, because that distinction is where most of the money is lost. Well established, with strong evidence for both lifespan and health span, not smoking, which remains the single largest modifiable factor. Physical activity, and here I want to emphasize something specific. Cardiorespiratory fitness is among the strongest predictors of all-cause mortality that has been measured. A large study of treadmill testing found that higher fitness was associated with progressively lower mortality with no upper threshold of benefit observed, and that the difference between the least fit and the most fit was comparable in magnitude to major disease risk factors. Fitness is not a vanity metric. Muscle mass and strength, which independently predict mortality and functional independence, and which are the difference between an old age you can navigate and one you cannot. Resistance training is consistently underprescribed. Sleep, adequate and regular. Managing blood pressure, glucose, and lipids, which is what our previous 4 episodes were about. Not drinking excessively. And social connection, which people are consistently surprised by. Meta-analyses have found that strong social relationships are associated with substantially reduced mortality risk, with effect sizes comparable to well-known physical risk factors. Loneliness is not merely unpleasant, it appears to be a genuine health exposure. Cognitive and social engagement, and continued learning, are associated with lower dementia risk. None of that is new, none of it is exciting, and all of it is what the evidence actually supports. Now the experimental territory, honestly. Caloric restriction extends lifespan reliably in many laboratory species. In humans, the calorie trial found metabolic improvements from moderate restriction over 2 years, but we have no lifespan data and never will, because the trial would take a lifetime. There are real trade-offs, including effects on bone density and muscle mass, and I want to say clearly that I am not recommending it. This is a research finding, not a protocol, and pursuing restrictive eating on the basis of animal studies is a poor trade for most people. Rapamycin extends lifespan in mice with unusual consistency. Human trials for aging are limited, it is an immunosuppressant with genuine risks, and dosing for this purpose is not established. Metformin, a common diabetes drug, has been associated in observational data with lower rates of several age-related conditions. A dedicated trial, TAME, has been proposed to test this directly. Observational associations in this area have repeatedly failed to survive proper trials, so this remains an open question. Senolytics are promising in animals and at an early stage in humans. NAD precursors, sold widely as supplements, have a plausible mechanism and so far, human trials have generally shown modest biochemical changes without demonstrated clinical benefit. And GLP-1 drugs are producing striking results for weight and cardiovascular outcomes, and whether that translates into effects on aging itself is an open question. The honest summary is that no intervention has been demonstrated to extend human lifespan. Not one. Everything sold on that basis is extrapolating from animals, from mechanisms, or from nothing at all. Which brings me to the industry, and I will be direct. Biological age tests based on epigenetic clocks are genuinely interesting research tools. As consumer products, they have substantial test-retest variability, meaning the same sample can produce noticeably different results, and their ability to guide individual decisions is limited. Treat a result as entertainment rather than information. Supplement stacks marketed for longevity are, almost without exception, unsupported by human outcome data. The resveratrol story is instructive, enormous excitement, extensive marketing, and a subsequent research record that largely failed to deliver. And a general filter that will serve you well. If someone is selling the product they are describing, apply the discount that deserves. If a claim rests on mouse data, remember that a great many interventions extend life in mice, and essentially none have been shown to do so in people. Which brings us to the reframing I want to end on. Almost nobody actually wants to be old for longer. What people want is to be well for longer, and then to decline briefly at the end. Researchers call that the compression of morbidity, and it is a far better goal than maximum lifespan. That distinction matters practically, because it changes what you optimize for. Cardiorespiratory fitness, muscle strength, balance, cognitive engagement, and metabolic health are not primarily about adding years. They are about the condition you are in during the years you get, and about whether the last decade of your life is one you can participate in. And on the biggest question, here is where the science leaves us. We do not age because a clock is counting down. We age because we are the products of a process that had no reason to build us to last, that funded maintenance for as long as it affected reproduction, took bargains that favored our youth at the expense of our age, and left the rest to accumulate. That is, oddly, the optimistic reading. There is no fundamental law being obeyed here. There is a maintenance budget that was never set for the lifespan we now actually live. Whether that budget can be renegotiated is the great open question of this field, and it will be answered by careful science over decades, not by anything currently for sale. In the meantime, the things that reliably work are available to everyone, cost very little, and have been known for years. That is unglamorous, and it is true, and the gap between those two facts is precisely where the longevity industry makes its money. That was today's question. If it was useful, please like and subscribe, because being intentional about your health is not one decision, it is a habit. And now the important part. This series exists to answer your questions. So tell us, in the comments, what you want to know. If it matters to you, it matters to someone else watching, and it deserves a proper answer. Ask us. We will do the work. Thank you for watching, and take care of yourself.